Today I will be dealing with the gland, which is primarily responsible for a major portion of the digestion of the food, the, the protein, fat, and carbohydrate. That is the pancreas. And this pancreas has both exocrine and endocrine, and it is the glandular part is the exocrine pancreas. Okay, so now this lecture I have outlined accordingly. Here in the beginning, in the first part, I will talk about physiology, anatomy, and histology of the pancreatic glands. Then I will deal with the functions of the pancreas. Subsequently, composition of pancreatic secretion. Try to emphasize on the functions of the components of the secretions. Then the method of enzyme secretion, the mechanism of HCO3 secretion, and the regulation of the exocrine function of the pancreatic secretion, both cephalic, gastric, and intestinal phases, and then pancreatic function tests. So these are the outlines of this lecture. And to start with, the pancreas is a mixed gland. It has both exocrine and endocrine secretions. Exocrine are those secretions which are coming through the ducts. The endocrine are those secretions which are entering into the blood. Maybe if I were to clarify that what is that exocrine and endocrine. So endocrine are those endogenous substances which are released into the blood and act at a distal site. Say for example, the pancreas, the endocrine part of the pancreas secretes insulin. Insulin is secreted from the beta cells of the pancreas, and this enters into the blood and activates the various tissues in the body, the liver, the fatty adipose tissue, and other tissues. Okay, so like that, it has endocrine secretions and exocrine secretion. Exocrine secretions are by the glands and the duct system. And these exocrine secretions contain enzymes which are necessary for the digestion of a protein, fat, carbohydrate, and even the nucleotides like DNA and RNA. So this also secretes the bicarbonate, copious amounts of bicarbonate in water that neutralize the acid chyme. It is controlled by the neurohumoral factors. Further, this is a pancreas. This is present in this C-shaped content or the duodenum. It's the duodenum. In the duodenum, this curvature of the duodenum, you have the pancreas, head of the pancreas is sitting, the tail of the pancreas hits the spleen here. And this has all these glands. These are glands. Glands are like this. These glands are empty into the ductules. These are the ductules and these ductal system. And these ductal system join to form the main duct. This the main duct that comes here and then joins with the bile duct. This is a bile duct. This is a gallbladder and this is a bile duct. So now this duct, this major duct is known as the pancreatic duct of Wirsung. This forms the major duct. Okay, so now this joins with the biliary duct. So this is then when it joins, this becomes the ampulla. This forms the ampulla of Vater, ampulla of Vater here. Then this ampulla, this is guarded by the sphincter, the circular muscles, circular smooth muscles, what is known as a sphincter OD. And this is released as and when required by the hormonal controls, especially the cholecystokinin and other hormones that regulate the contraction and relaxation, including and in addition to other hormones. Sometimes in some individuals, this duct system, pancreas has an additional duct system that opens a little higher up in the duodenum. This is a variation. Just I want to say that it has a glands. The glands are similar to the salivary glands, and all these glands have a ductal system. They join and form the pancreatic duct, major pancreatic duct, what is known as a duct of Wirsung. and this duct joins with a common bile duct. This is a common bile duct. So this opening here is a ample of water, and there may be an additional duct. This is about this thing, and this is a acinar gland, and this acinar gland secretes enzymes here. These are mainly the zymogen granule situated, and this secretes the enzymes. The duct system, the greenish one, that secretes the bicarbonate and water. Now coming back here, come next, what are the functions of the pancreas? The pancreas secretes the bicarbonate and water so that it neutralizes the acid chyme coming from the stomach. That is the number one. Then by neutralizing, it provides the optimum pH for the activation of the intestinal enzymes and the digestion and absorption of the various components in the intestine. And it will facilitate the exchange of bile and other constituents present in the intestine. And it has an enzyme system for the digestion of protein. It has an enzyme system for the digestion of the fat. That means it participates in protein digestion, it participates in fat digestion, it participates in carbohydrate digestion. Protein digestion by the trypsin, chymotrypsin, carboxypeptidase, and other peptidases. The fat digestion is by the lipases. The carbohydrate digestion is from the pancreatic amylase, and DNA and RNA digestion, that is DNase and RNase. Collagen and elastic tissue digestion by collagenase and elastase enzymes. And in addition to this, it prevents the secretion from the glands, prevent the digestion of the glands itself. That means there is another enzyme system that is a trypsin inhibitor is synthesized from the glands that prevents the action of a trypsin on the glands and the surrounding tissue. These are the exocrine functions of the pancreas. Let us move further. What is this pancreatic juice? Nearly we have about 1.5 liters of pancreatic juice secreted per day. And this secretion varies with the stages of digestion. Maybe in the resting stage, it may be slow. And as we consume food, it increases. As the food reaches the duodenum, it reaches further. So that is the peak. Maybe I will show you that. Though, and the pancreatic juice is alkaline. So it may be the pH is around 8. And it is rich in bicarbonate ions and also enormous amount of water associated with it. So if you are looking at the bicarbonate ions, it is about 145 milliequivalents as against 24 milliequivalents in plasma. It's nearly, you can just see that 6 times the plasma concentration. The bile and intestinal juices are neutral or alkaline, and these secretions neutralize the gastric acid. So that is the main purpose, plus provide the suitable environment. By the time the chyme reaches the jejunum, the pH is neutral. So that means it becomes around neutral is 7. What are the compositions? Again, I repeat the composition of the pancreatic juice. So you have this, the daily is 1.5 liters. It has water, it has solids. Solids, we have the inorganic and organic. Inorganic are the cations and anions. These cations are sodium, potassium, magnesium. The pH is 8. And the anions are HCO3, large amount, chloride, HPO4, and sulfates. These are the anions. Then we have organic molecules. It contains the proteolytic enzymes, lipolytic enzymes, amylolytic enzymes. Then it has a nucleotidases, that is a DNase and RNase, and elastase and collagenase. These are all enzyme systems. They will digest. And it has an additional enzyme system which prevent the proteolytic actions of the trypsin, that is a trypsin inhibitor. It also secretes mucin. These two are protective in nature. Then if you are looking at this the composition, because I have broadly classified these composition, now I will try to give a little bit of detailed outline of these enzymes. What are these proteolytic enzymes? The proteolytic enzymes are here the trypsin, chymotrypsin, carboxypeptidase. The trypsin activates proteins to peptides. That means it breaks down the proteins to peptides. Peptides are a group of amino acids. It's not complete digestion up to the peptide level, and the peptidases are there. Peptidases are there, maybe carboxypeptidase that will digest the peptides to amino acids, and peptidases are there in the intestinal juice. The chymotrypsin, again, it will also act on proteins up to the level of peptides. Carboxypeptidase that acts on the peptidases, carboxy terminal of the peptidase, and that will digest up to the amino acids. So this is about a proteolytic activity, trypsin, chymotrypsin, and carboxypeptidase. So now, usually these trypsin, chymotrypsin, and carboxypeptidases are not there in the native form as they are. They are either in the trypsinogen, chymotrypsinogen, or procarboxypeptidase, something like that. So that they are in a precursor of these things, and they are activated in turn by the trypsin. The beginning is started by the trypsin. Then we have amylolytic, that is the carbohydrate digestion. So this is by the pancreatic amylase. You remember the salivary amylase has started the starch or the carbohydrate digestion. Here from the pancreas, it will act, or no, it will complete the almost the digestion here. So that means the starch and other carbohydrates, and including a glycogen and other carbohydrates, or amylase acts upon, because there is sufficient time, so good pH, optimum pH, and all the environment, and it will digest. Especially this environment is provided in the first part of the duodenum and in the intestine, initial segment of the intestine. So they will be converted in up to the level of the di and trisaccharides. Now these di and trisaccharides are converted to monosaccharides by the monosaccharidases present in the intestinal juice. So that is amylolytic. They will make it to the level of the disaccharide or a trisaccharide. Lipolytic enzymes. The pancreatic lipase is one of those major enzyme which digest the lipids. That is, a neutral lipid is converted or digested up to the level of fatty acid and to the level of the monoglyceride. And it has a cholesterol esterase, and this cholesterol, it converts the cholesterol ester to the cholesterol. Phospholipases, it will digest the phospholipids to fatty acids and phosphate. And we have a nucleotidase, that these are RNAs that will split into the particular RNA molecules or RNA constituents, DNA, that whatever the CGA or whatever the pattern of the nucleotides, so they will be released. So the formation of the nucleotides from these RNAs and DNAs. In this table, I have given a little bit detailed this thing. This table I have taken it from Genog. So now I just quickly read this table. Trypsin is in the form of a trypsinogen. This is activated by the enteropeptidase. That means this entero is intestinal peptidase, because the trypsinogen enters there, that will activate the trypsin. Then it will act on the proteins and polypeptides, and that would form up to the level of basic amino acids, arginine and lysine, and some peptide bonds. So it will form up to that level. Where exactly it acts, that is mentioned, to cleave a peptide bonds on the carboxyl side and basic amino acids. That is where the site of action of the trypsin. A chymotrypsin, it is present in the chymotrypsinogen, and it is activated by the trypsin, because now the trypsin is taking the role, and this will act on the proteins and polypeptides. And where it acts, it cleaves the peptide bonds on the carboxyl side of the aromatic amino acids. Here, the basic amino acids, if you are looking at basic amino acids, the aromatic amino acids, that is the amino acids with the benzene ring. Then elastase, it is a pro-elastase. It is also activated by trypsin, elastin tissue, elastin molecule, and some other proteins. Cleaves the bonds on the carboxyl side of the aliphatic amino acids. So if you are looking at here, aromatic, and this is aliphatic amino acid. Carboxypeptidase, it is again exists in the form of a procarboxypeptidase activated by trypsin, and again on the proteins and polypeptides, and cleaves or act at the carboxyl terminal of amino acid that have aromatic or branched aliphatic side chains, aromatic or branched aliphatic side chains. So these are some of the biochemical terminologies you may like to be familiar with that. Carboxypeptidase B, again as a procarboxypeptidase B, activated by trypsin, and it cleaves carboxyl terminal amino acids in the basic side chains. So this is a have aromatic or a branched side chains and a basic side chains. Colipase, it exists as a pro-colipase, again activated by trypsin. It acts on the fat droplet, binds to pancreatic lipase to ile droplet in the presence of bile acids. So because now it will take help of the bile or bile salts. These bile acids, they will try to digest these fatty droplets. The pancreatic lipase, there are no activators there because maybe bile will be helping that and making up for the, it acts on the triglycerides, convert into monoglycerides and fatty acids. Cholesterol ester hydroxylase, so it will act on the cholesterol esters to make the cholesterol. Pancreatic amylase, it is activated by the chloride ions, and acts on the starch, and it is converted to the disaccharides or monosaccharides. Ribonuclease, RNA nucleotides, DNAs, so DNA nucleotides, phospholipase A2 activated by trypsin, and phospholipids, fatty acids, and lysophospholipids. So this is in general about the overall the actions of various enzymes of the pancreas. Now come back here, how the enzyme secretion is taking place in the pancreas. This is one of the typical acinar gland or a typical acinar gland here. This is the basal side in which it is associated with the blood vessel. This is the basement membrane, the blood vessel. And it receives the nerve fiber. The nerve fiber is from the vagus. And it has this rough endoplasmic reticulum. You can see this rough endoplasmic reticulum and a lot of mitochondria. So they will secrete these through the Golgi apparatus. These are vesicles or zymogen granules. These are zymogen containing. These are granules there. The zymogen granules are located here in the apical portion. This is the apical portion of the cell. And these are the Golgi complexes. And these are zymogen granules are nothing but the vesicles filled up with the enzymes, various enzymes we have listed. So this is granules containing the digestive enzymes, what is called the zymogen granules, are formed in the cell and discharged by the exocytosis into the lumen. So these are the zymogen granules. Here you just see the exocytosis breaking down of this vesicle. So this is released into the lumen. This is the duct. So now you have the various structures. So this is where by the stimulation of the vagus, acetylcholine is released. Acetylcholine activates the cellular production of this whatever the enzyme system through the intra-adenine cyclase or cyclic AMP mechanism, and that would bring about the formation of the zymogen granules. And also because of the arrival of the impulse, that would release the granules from this thing by the exocytosis. So now this is activated by the acetylcholine. I mentioned the vagus or acetylcholine. This secretion, either even a production on this side or the secretion on this side, is activated by the vagus and by the cholecystokinin, a hormone secreted from the duodenum. And this action here, sometimes some leak in the trypsin will activate the trypsin itself. And once it is activated, it's a very potent enzyme system that digests the cells itself, any molecule. So that is why there is also, along with this, there is another substance which is released, a trypsin inhibitor. So that means this is been having a smear of the trypsin inhibitor, so that even by mistake the zymogen granules releasing a trypsin will be a disaster that may lead to a severe condition known as a pancreatitis. Now this is about the enzyme secretion. Now I'm just trying to make you feel about the importance of the trypsin inhibitor. A trypsin inhibitor prevents the digestion of the pancreas itself. This is a very important enzyme. And this is secreted by the same gland which secretes the trypsin. The proteolytic enzymes of the pancreatic juice not become activated until secreted into the intestine. Suppose if they are activated here, they start digesting, because whatever the tissue or the cell is made up of the protein, fat, or carbohydrate, whatever. So they start digesting. The same cell that secrete proteolytic enzymes into the acini of the pancreas secrete simultaneously another substance called trypsin inhibitor. It is an enzyme. This substance formed in the cytoplasm of the glandular cells, and it prevents the activation of a trypsin both inside the secretory cells and outside. Say, for example, here in the cytoplasm, you have this trypsin inhibitor. It prevents the action of a trypsin here. By mistake, some of the zymogen granules break down here. That would be a disaster. And here in the lumen, that also it prevents. And because of it, is a trypsin activates other pancreatic. See, look at the trypsin. If you are looking at the chart, trypsin activates the number of proteolytic enzymes. That is why this is an important substance to prevent the action of trypsin. Now, this is about the HCO3 secretion from the pancreatic cells. So we have the duct of the lumen, and this is a basolateral portion where the blood is there. So now the reaction starts with the what is that? The carbon dioxide, because this is a cell which is metabolically active, and this carbon dioxide combines with water and is enhanced by the carbonic anhydrase, which forms the HCO3. Now this HCO3 is co-transported with the exchanger. There is a chloride bicarbonate exchanger. This HCO3 is taken up and it is transported into the lumen, and that's exchanged with the chloride. So that means the chloride enters, it takes the chloride and the bicarbonate, they are exchanged, and this is a chloride bicarbonate exchanger system. This is the first one. Then comes here the H ions which are made, which are because the bicarbonate is moving this side and H ions become not this thing. Now these H ions are pumped into the blood. This is the basolateral portion, this is blood, and this is being by the sodium hydrogen exchanger. Sodium hydrogen exchanger. This is NHE1. So that means these H ions are exchanged for sodium. So now another important component, there is a sodium bicarbonate co-transporter, because some of the bicarbonate present in the blood is also transported, because the bicarbonate levels are decreasing, and this bicarbonate levels, these bicarbonates are pumped into the cell, and which in turn further used for this exchange system. That means bicarbonate are replenished by this sodium bicarbonate co-transporter system. Now, this is sodium is recycled because the sodium is entering inside the cell. It is sodium is recycled by sodium potassium ATPase. This is sodium potassium ATPase pump. Now the sodium is coming back recycled into the blood, and for that potassium is exchanged. And the potassium is trying to move into the blood by the potassium channel, but it's again exchanged. Now, so now this chloride ions, sometimes we have the cyclic AMP will be activated, and the chloride ions will be transported because there will be a large amount of potassium ions. The relatively more amount of potassium, the positive ions accumulate in the cell, and to neutralize that, there is what is called a CFT or channel, chloride channel, and this chloride channel is this is the cystic fibrosis transmembrane receptor, transport receptor, cystic fibrosis transmembrane receptor, and this will transport the chloride ion, and this chloride ion is pumped in because there is, and it is dependent upon cyclic AMP. This is the overall production of the HCO3 in the pancreatic duct system. So that means the enzymes are secreted in the acini, and the bicarbonate is secreted is in the duct system, and because of this bicarbonate, the chloride and some amount of sodium that enters in the hyperosmolality, and that would increase the osmolality, and that will pull the water by osmosis. If I were to tell the first step is the production of the bicarbonic acid from CO2, and that would dissociate into H and HCO3. Then HCO3 is co-transported with the sodium on the, or the HCO3 is co-transported, and then there will co-transported on the other side. There is some HCO3 is coming from the sodium co-transport mechanism. Then the HCO3 is exchanged for a chloride, and the chloride is recycled, and the sodium H ions are exchanged for sodium and water movement. That's what I described all of them. This one I have taken it from Genang. Now, coming back, what are the various factors that regulate the pancreatic secretion? It is controlled by both the nervous and hormonal factors. So these nervous and hormonal factors are involved with three different phases. The three different phases, as we have seen with the gastric secretion, that is the cephalic phase, gastric phase, and intestinal phase. So the nervous mechanism is through the vagus. Vagus supplies the gland. The vagus supplies the gland, and vagus releases acetylcholine. And it is through this acetylcholinergic action that would increase the synthesis of the production, because this is through the M2 receptor, and also it will enhance the release of the zymogen granules. So then that is about the vagus. Now vagus alone will not be able to produce all the effect that is required, because there is so much amount of enzyme system is required for the digestion of our food. For that reason, there will be hormone. There will be another hormone. That hormone is a cholecystokinin. Cholecystokinin will help the vagus for activating the glandular cell. This is a hormone secreted from the duodenum, and from there it reaches the gland system via the blood and activate. So that means vagus and CCK together, they increase the enzyme synthesis, and CCK 80%, the vagus action 20%. You just see this is the large amount of these things. Then we have another hormone here, and this hormone adds water and bicarbonate from the duct system, because whatever is secreted in the gland has to be evacuated through the duct system. What this duct system is doing, it is flushing. So the whatever the enzymes coming here, and these enzymes are pushed from the water and bicarbonate. So that is done by the secretin. That's a hormone secreted from duodenum. Now I have just summarized here. Acetylcholine and CCK, cholecystokinin, produce enzyme secretion from the glands. The secretin activates the duct system to secrete the water and bicarbonate. That is the basic regulation, and we see this whole thing is being placed here. So acid in the stomach releases secretin from the. So maybe I will just come back here. So this one here, cephalic phase, gastric phase, and intestinal phase. Now I talk about the cephalic phase. The cephalic phase of pancreatic secretion, as happened with the stomach, it will start with even the conditioned and unconditioned reflexes. The conditioned reflexes involves the mastication, swallowing, the gastric contractions, and all those things. They will be the unconditioned reflexes. The conditioned reflexes involve the sight, smell, thought, and the taste of food. And these conditioned reflexes are initiated by the cerebral cortex, the prefrontal cortex, the limbic system, amygdala, and it is mediated through the hypothalamus. From the hypothalamus, it is active dorsal motor nucleus of the vagus is activated, and that would reach the pancreas. That is how the cephalic phase is taking place. This phase normally amounts for about 20% of the pancreatic secretion. Please make this is not a gastric secretion. 20% of the pancreatic secretion. Moderate amount of enzyme secretion, very moderate amount of enzyme secretion, but a little amount of flows as a small amount of water and bicarbonate are secreted, waiting for a secretin response. Because of this is purely a neural component. Neural component drives nearly 20% of the enzyme secretion, and the water secretion is not activated. The water secretion is exclusively activated by the hormone secretin. That is about the cephalic phase. Now, the gastric phase has two components. One, the neural stimulation started by the cephalic phase now will be continued because of the mechanical or chemical participation of the gastric. That will be the inputs are going through the vagal nerve, reaching to the nucleus tractus solitari, and then to the dorsal motor nucleus of the vagus, and then to the vagal efferents. So that means this is initiated by the stomach itself. The mechanical distension of the stomach or the chemical components of the stomach that will stimulate the vagus, and that would further make the vagal efferent activity. Now, the second component is a local enteric reflexes. These local enteric reflexes are generated by the myenteric plexus in the stomach. They will activate the gastroduodenal myenteric neurons, and that would activate. So that is the second part in which the gastric phase is continuing. Maybe this will only account, the gastric phase is only account for 5 to 10% of the total secretion, because it does not make a very large quantity of secretion. And again, the ductular system, the bicarbonate water secretion is dependent upon the secretion action. In the intestinal phase, the entry of chyme in the duodenum, and in the initial segment of the small intestine, that would secrete two hormones, secretin and cholecystokinin. Secretin is by the S cells of the duodenum, and this secretin in turn secretes the copious amounts of the bicarbonate and then water. So you can just see that these bicarbonate that will neutralize the HCl coming from the HCl present in the chyme. Chyme is the product of the stomach or gastric digestion. So then HCl from the stomach and the bicarbonate from the duct system, that would form NaCl and H2CO3. Now this H2CO3 is dissociate into carbon dioxide and water. So that is how the hydrochloric acid is neutralized. Hydrochloric acid from the stomach is neutralized from the pancreatic secretion. Now we have a second hormone, CCK, cholecystokinin. Previously it was known as a pancreozymin, and now we know that it is the same molecule. Now in between something it was named as a CCK-PZ. Now it is clearly mentioned as a cholecystokinin. And it is activated by the presence of food in the duodenum, especially partially digested protein products that stimulate the eye cells. You just see that eye cells in the duodenum to secrete CCK. CCK via the blood reach the pancreatic glandular cells and stimulate the pancreatic glandular cells to increase the synthesis of enzymes. This is about 70 to 80%. 80% of the enzyme. 20% I mentioned is activated by the nerve, that is the vagus. So 80% is by the CCK. And so in addition, the cholecystokinin as the name indicates, the cholecystokinin. Cholecysto is a gallbladder. Kinin is a humoral substance. Then this will stimulate the gallbladder contraction. So that is the name. If you are talking about cholecystokinin, that means it also produces the gallbladder contraction, so that the contents of the gallbladder are released. If you are looking at the pancreatic duct and the common bile duct, they join together, and both of them, when they are formed, they are released into the first part of the duodenum. So that means the pancreatic juice comes along with the bile. Now, here is one of those things, how these two hormones are operating. Here, the presence of HCl in the duodenum, you see that pancreatic secretion. This is water and bicarbonate. See that it is maximum amount of water. Bicarbonate is produced, and enzymes are very little. And the soap is a fatty this thing, and you have both water and bicarbonate as well as enzyme, because you require lipases. Then we have peptones, that is the peptide molecules, the partially digested proteins. So what do you see? You see the secretion of this water and bicarbonate is less, but secretion of the enzyme is very high. So this is how, that means 3 parts. One is the HCl alone, increasing only bicarbonate and water. Then it is a fatty thing, that are in the sense, in the fatty meal we have both of them are equally participating, that is the enzymes as well as the bicarbonate. In case of the peptides or peptones, it is mostly enzymes, not the bicarbonate and water. Now come back here. What happens when you are single dose of secretin on the composition and volume of the pancreatic juice in human beings? Here the secretin is given, and this is the unit, the 12.5 units of intravenous secretin is given here. And what happens, you just see that the bicarbonate level, this is the resting this is the time, the basal activity, and this basal activity suddenly jumps, whatever the level, it goes up to 150. So you can just, there is a peaking here within 10 minutes, and then it is coming back. At the same time, these chloride ions are decreasing, because there is a exchanger, no, chloride and bicarbonate exchanger is there. So for that reason, chloride ions come down, just in the opposite. And the amylase activity decreases, because this is adding up the water, and maybe because of its dilution effect, the potassium remains unaltered. So this is the effects of the secretion. You can just see that secretion is doing this. So I mentioned about, so that means the CCK increases the enzymes, secretin increases the HCO3. Now coming back, the pancreatitis, it's an acute condition. It is an acute emergency condition. Suppose if it is not treated in an emergency situation, the patient will die, because in this pancreatitis there will be a release of the gastric or the pancreatic contents are released into the peritoneum and surrounding tissue. And if these pancreatic contents are released, they contain enzymes which can digest anything and everything. So because of that, this is an emergency situation, and the immediate surgery is required to clean up this mess. So now just try to see that when the pancreas becomes severely damaged, or when a duct becomes blocked, pancreatic duct, large quantities of pancreatic secretion sometimes become pooled in the damaged area of the pancreas. Then what happens under these circumstances? The effect of trypsin inhibitor is often overwhelmed, because it will not be able to prevent. It's not able to prevent the action of a trypsin, in which case the pancreatic secretions rapidly become activated and literally digest the entire pancreas, not only the pancreas, the surrounding tissue, within few hours, giving rise to the condition called acute pancreatitis. Very severe condition. It is treatable condition. The patient can recover, but only thing that immediately he has to be taken to the operation theater, and this has to be cleaned up. This sometimes is lethal, because accompanying circulatory shock and other effects they will produce. If not lethal, it will lead to lifetime pancreatic insufficiency. This is about pancreatitis. Now coming back, I just list these pancreatic function tests. So there are two types of tests: the direct tests, indirect tests. In the indirect tests, that requires intubation, and that do not require intubation. What are those direct tests? The direct tests are those secretin response test. You inject secretin, see bicarbonate response. That means you have to take the contents of the, you have to collect the contents of the duodenum and trying to analyze that. So that is what you inject the secretin, and you see the contents in relation to time, as it was done here. It is a secretin test. Secretin test, wherein secretin was injected, and you are estimating the secretin level. So like that, CCK test. In that, CCK response is the enzyme. So that means you start measuring the enzymes present in the duodenal contents. That means you have to intubate. This is the direct test. And sometimes both CCK and secretin are simultaneously given, so that whether the glandular as well as the ductal system are operating synchronously. So that is a direct test. Indirect test that require intubation. So this is one of those tests, wherein a Lund test, wherein the trypsin and lipase levels after intubation, trypsin and lipase levels after oral test meal. So that means you give the test meal containing 6% fat, 5% protein, and 15% carbohydrate. So this orally you allow him to swallow, and then after this ingestion, so you try to intubate, collect the duodenal contents, and look for a trypsin and lipase levels, how they are operating, because here you are looking at the fat, you are looking at the protein. This is one test. This is a Lund test. And we have a small intestinal infusion test. So that means here what you do is you infuse hydrochloric acid or amino acid in the jejunum, because the infusion of HCl activate the HCO3 release. Infusion of amino acid activate the release of the enzymes. So now after infusing, and see their responsiveness, whether you are getting the bicarbonate or the enzymes. So that is the small intestinal infusion tests. These are indirect tests, and these require intubation. And there are other indirect tests, they without intubation. Just it is the estimate the fecal fat content. So that means 24-hour fecal material is collected and the fat content. If the fat content is more, that means the lipase activity is less. So that is an indirect. This is an indirect indication that does not speak on only one aspect of the pancreatic thing. So then, because that is also dependent upon the what I will say the bile salts, because bile salts are also required. So for that reason, fecal fat test has its own limitation. The fecal chymotrypsin test. So again, the fecal content for 24-hour is this thing, and the chymotrypsin level is estimated. And if the chymotrypsin is there, so that means that was that is not, the enzyme is not utilized. So there is another test, what is called a NBT para-amino benzoic acid test. So this is NBT is a N-benzoyl L-tyrosyl para-amino benzoic acid. And this one, when activated by the chymotrypsin, is converted into para-amino benzoic acid. So that means you take the duodenal contents and this particular substrate, then you add this particular substrate. So that means then if it is converting to para-amino benzoic acid, so that means chymotrypsin is present. So this is the basis or the principle on which this NBT-PABA test is measured. So then we have a pancreolorel test using a fluorescein bindings and 13 carbon mixed triglyceride breath test. This is a radio linked test. These are of a little higher up, and you can just look into these things in the textbooks. So basically, you have the pancreatic function tests that utilizes either direct or indirect methods, and these are the methods in which you can just indirectly see without any intervention. Now, thus the pancreas secretes the enzyme systems necessary for the digestion of the protein, fat, and carbohydrate. It is also necessary for the digestion of the nucleotides, and it is necessary for the neutralization of the acid from the stomach, and it provides it try to set in the pH of the intestine. So these are the main pancreatic functions, and it is being released from this gland, and there will be the enzyme secretion, and there will be from the gland, and the bicarbonate secretion from the duct. So that is the in total about the pancreatic secretion. Now these are the textbooks I referred upon, the Guyton and Kenong's textbook and Barn and Levy. And in the next class, that's on Monday, we will talk about bile secretion and bile functions. In the next class, I will continue with bile secretion and bile functions. Okay. Meanwhile, I am giving you some questions just for your home assignment. So this is a full question. Describe the composition and the functions of pancreatic secretion, and add a note on regulation of pancreatic secretion. This is a full question, full essay type of question. Or we have a short questions here. I have just mentioned short questions which are there. All these short questions are answered in my lecture. Secretin, cholecystokinin, the composition of pancreatic juice, the mechanism of HCO3 secretion. Please make correct, HCO3 secretion, and acute pancreatitis. I mentioned about that. Trypsin inhibitor, how it is important. Enzymatic secretions of the pancreas, and pancreatic amylase, and the pancreatic function tests. Please work on these things try to make a note of these things immediately after putting 1 or 2 points. That would help.